approximately 2 liters a day, all right? Minimum value should be 30 mils an hour, all right? So at the very minimum, you should be making at least 30 mils an hour, approximately a cup in 8 hours. 240 mils is the cup. All right, so color has to deal with concentration. So the less urine you produce, because it's more concentrated, the darker the color is going to get. So the best color is one or two. Okay, a pale straw color, very, very pale. If you look over on the counter, you can see that either those two over on this side or those two over on that side, that kind of clear yellow, all right? So you should barely be able to detect it in the bottom of the urine bowl. Now you probably notice if it's really, really, really dark, it sinks to the bottom. It doesn't disperse very quickly through the water of the toilet bowl. And that's because the darker it is, the more concentrated it is. We've removed water, so there's more particles per volume, okay? We have a higher density, so it's going to sink. Just like if you take the ocean water with all the salt, it's easier to float because all those salt particles are floating, okay? Turbidity has to do with cloudiness. So here you can see the graph lines. Here you cannot. So this is cloudy and this is clear. Doesn't mean it doesn't have to have any color. It can still have color and be clear. When it's cloudy, it's usually because of bacteria or cells. So if you have a cell sloughing off the lining of your bladder, or you have a lot of tiny little casts, or there's bacteria, you have a urinary tract infection, okay, it's going to have some component of cloudiness to it. So that's what turbidity is. Odor. So some people, not everybody, but a lot, have an enzyme that if you eat asparagus, converts it to a specific odor that's detectable, okay? Pretty quickly, within like 15 minutes or so after you eat it. If you go to the bathroom, you can smell it in your urine if you have that enzyme. Also, when urine is exposed to air, more of the ammonia ion is formed, and so it has that, old baby diaper smell to it. Or we have a lot of feral cats around here, so you walk through the courtyard in our offices and you can smell their pee amongst the plants. That's because of the ammonia. pH. Urine has a wide range of pH because it has a lot of buffers in it that we'll talk about on Tuesday to bind to the hydrogen ions. So it could go as low as 5. We don't want it to get down to 1 to 2 or it would really burn. It's like hydrochloric acid. So it has a lot of buffers in it. It can go up to 8, but usually it's between 5 and 6. Okay? So slightly more acidic. Our blood is about 7.4, 7 being neutral. Specific gravity. That's what you see over here on the table. These little cylinders are what's called a urinometer. What does specific gravity have to do? How is it related to concentration? It's a comparison of density of mass to volume density, all right? So density, if we take the volume of something, so pure water, we have 1.0 mils of water, and it doesn't have minerals dissolved in it, it's going to weigh 1 gram. So it would have a specific gravity of what? Okay? Now, nobody ever makes urine that's completely pure water, all right? So we would never expect urine to have a specific gravity of less than 1. The more concentrated it is, the more urea and ammonium and sodium ions it has in it. So the higher the specific gravity. So specific gravity greater than 1 is indicative of how concentrated it is. Those go hand in hand. All right? So very, very dark urine is going to have a very high specific gravity. So the normal range is 1.003 to 1.03. This would be a lower specific gravity. This would be the higher specific gravity. All right? So the urinometer is floating in this urine. And if it has a high specific gravity, is it going to be sinking down or floating higher? Floating higher, because all those particles are helping to float the urinometer. All right? So you probably heard if you go over to the Red Sea, which is very, very, very salty, you can't sink. You're like, 3 inches up higher in this ocean than you would be in normal water. You're gonna sink it's easier to sink in a swimming pool than it is in a regular ocean because of the amount of salt that's in there, and the Red Sea has even more. So then our uranometer is going to be higher. You can see the number here is 1.000, 1.020, 1.030, 1.040. Okay. Now, whenever we're reading something like this, it's a little difficult because you have to read the meniscus. The meniscus to the surface tension means that if we have a tube, the water is going to get pulled up the sides of the tube, all right? So you want to estimate where this is over here. So when you look at the uranometers over there, they're going to have a line with markings, and there'll be a 1.0 and a 10 and a 20 and a 30. So if it's at the 10, you're going to read this as 1.010. Okay? If you're here in the middle, it would be 1.005. If you're down here, it would be 1.018. And then when it got to here, it would be 1.020. Okay? So 1.03 is our maximum normal specific gravity. Higher than that, it's really, really, really got stuff in it. Okay? All right. So in your lab packet is a list of things that are normal and abnormal. There's no place to write the results of this since I'm putting this as an online lab. So what we're going to do for the activity is 2 parts. One is to look at the specific gravity on those uranometers because I want you to be able to know what it looks like, but you can read specific gravity on here. Okay? Those of you that have worked in hospitals or nursing homes are familiar with dipsticks. And so there are boxes over here that contain a rack and a pen to mark the numbers 1, 2, 3, 4. And then test tubes. And you need 4. All right? And this is fake urine. So urines 1 and 2 are on this side, 3 and 4 are on the other side. So I just want you to take a look at the specific gravity ones, but you're going to read your actual numbers. I want you to be familiar with what the setup was. So if you're not familiar with this, you want to be able, when you're going to read it, you're going to hold your dipstick like this, okay? And the very top, there's little indicators on here that change color after you dip it in the urine. So the very first one you're going to read here is at the top of the arrow is the number of white blood cells, or leukocytes, all right? And then then it's nitrogen, which is indicative of infections, protein, which we don't want unless you've got a barrier that's destroyed in the filtration membrane, pH, blood, specific gravity, SG is specific gravity, and ketones, which is indicative of breaking down fats, and glucose. Okay? So those are indicated by the little colored squares. So what you want to do is fill this up about a little bit past halfway. I still need to use this for Monday night. Okay, so we don't want to use it all. And then dip this in, and you can tilt this a little bit to get it all wet. Tap it against the side, and then it wants you to wait anywhere from 2 minutes to 60 seconds to read it, and then take a picture is what I would do, okay, rather than write down the data. Just take it over there like I did, lay it down, and take a picture. And then on the online lab report, I'll give you different views, and I want you to be able to read across here. So this one is showing that there is nitrates present, not nitrogen, nitrates present, a negative to a trace of protein, pH of 5, and a ton of blood, even more blood than a large amount, because I put in 2 drops of sheep blood. Specific gravity is about 1.010, no ketones, and negative for glucose. Okay? So on the online lab report, I'll have, you have this on the, in color, but I'll have a read chart like this and a dipstick, and I'll ask you to read it, okay? Or I'll ask for the results of urines 1, 2, 3, and 4. So take a picture and be able to, mark down which one it is, and that's essentially our lab, okay? But I want you to be able to interpret a dipstick. I want you to know what these different terms are that we use to testify, not testify, but to test the urine.
Are we
Okay?
doing all four?
Yes. You're gonna do all 4. Alrighty? When you clean your test tubes, you don't have to take this to the bathroom. You can just pour the fluids down the sink. Okay? And if you've used the Sharpie to number the test tubes, there's alcohol over there. If you would help the lab techs off by using the alcohol to rub off the Sharpie.